This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 176, recorded on March 22nd, 2012. Hi, everybody. I'm Vincent Racaniello, and you're listening to TWiV, the podcast all about viruses. Joining me today from North Central Florida, Rich Condit.
Hi there, Vincent. How are you?
I haven't spoken to you for a while, have we?
No, I've been out messing around.
2 weeks, is it?
I think so. Week 1, I was on a sailing trip, and week 2, I was on a grandchildren's skiing trip.
No, week 1 you were here because the sailing trip was cut short, remember?
Ah, that's right.
Yeah.
That's right. Yes, okay, so I've only missed a week. Okay.
Well, we missed you. It seems like you've been gone forever.
It feels like I've been gone forever.
Good to have you back.
Yeah,
hey,
great to be here.
Is it in the 80s down there?
It is 84 degrees, and we got these sort of springtime/summertime big white, I guess they're cumulonimbus or something clouds. They're just all stacked up on each other. They're beautiful. It's been gorgeous here, and it's just wonderful.
It's really been unseasonably warm here. It's 24 degrees centigrade or Celsius, as I've been told.
Celsius.
Also joining us today from Western Massachusetts, Alan Dove.
Good to be here.
How are you doing?
Doing great. It's 82 Fahrenheit, 28 Celsius up here.
28 Celsius.
Yeah.
Oh my gosh, why is it so warm there?
I don't know.
I would think New York City would always be a few degrees warmer.
Yeah, you ought to be getting the heat island effect, but maybe the coastal breezes are cooling you down.
Neat. Oh, all right. Well, the crocuses are past. We've got other things popping through the ground now. It's so warm. And, of course, here in New York, they haven't turned on the AC yet. So it's pretty hot. We have a couple of observers here in studio today who I would just want to mention. We have Danielle and Natalia, who are virologists here in New York City, and they are going to be starting a virology podcast in Spanish very soon. Is that right? So why don't you say hello, Danielle, and tell us what you're going to do exactly.
Hello, everyone. I'm Daniel Blanco, and I want to let you all know of all the Spanish speakers out there about a virology podcast in Spanish. Soy parte de un entusiasmado grupo de gente que planea realizar un podcast sobre virología para la comunidad de habla hispana. Así que estén pendientes, por favor.
And will it be esta semana en virología?
Tentativamente, sí.
He thinks yes.
Okay.
They're working on a title. I told them they didn't have to do that, but they could come up with something else. But we'll help them and advertise for them and help them get going, because I think that's a great idea, right?
Yeah.
They can reach a lot more people. And it's just a matter of having time, right?
Yes, but...
Making time.
Making time.
Making time
to do it. So they're going to watch us today, how we do this, and listen in, and they're here right in studio. So I hope that'll help out. All right, today we have an all-email episode. And before we actually get to the specific emails that we're going to be doing, there are a couple of follow-ups from the most recent TWiVs that I want to just go through. On TWiV 175, which was last week, More Than One Way to Skin a Virus, we talked about a paper where they use Q dots. And if you go over to the TWiV.tv posting for that episode, there's some interesting information and comments about Q dots, which are ways to label material so that you can see them in a fluorescent microscope. You don't use Q dots, do you, Rich?
No.
Okay.
But I'm going to have to look into this. I'm just looking it up now.
Just don't clean your ears out with them.
So what are the advantages of Q dots?
They're very bright, and you can have multicolors. But as you see on the website, sometimes they're too bright, and they don't work well for confocal microscopy.
Can I hook them up with antibodies and pick out a specific protein?
Yeah,
or
you can hook them up with anything pretty much, yeah.
Okay.
So Richard wrote about this. Just listen to this week's TWiV, and the Q dot dyes you mentioned are also used in electronics, There they're used as an ultra-precise phosphor. In that application, blue light from LEDs can be re-emitted as red and green. This gives an ultra-precise RGB light source, allowing highly accurate color rendition on LED backlit TVs. It's surprising how often these crossover applications crop up. Another good reason to listen to TWiV. This should happen more and more as the technology singularity approaches. That's great. Thanks, as always, for the interesting podcast.
So is there some like time out there where all of technology is going to be merged into a single thing? Is that the idea? I'm not sure I want to be at the center of this. Or maybe I do.
The singularity is when men and computers reach the same point, right?
Ah, okay.
Is that right, Alan?
Yeah, I think that's the idea.
So
when the computers become as intelligent as humans.
Yeah. And so I guess in technology, all technology comes together. Yeah.
Okay.
So Richard has a few comments about magnetotactic bacteria, which have to do with a TWiV episode. So I won't read them here. But he says, PPS best coffee maker, Aerobe AeroPress. Simple but highly effective. Hey, maybe we've started something here.
Yes, we this is in addition to the weather, now we're doing coffee makers. Did you look this up?
Yeah, it's a cool looking coffee maker. I have a coffee maker already, so I don't need any more.
Yeah.
So this is it's like a French press.
Yeah.
Okay.
Yeah, it's a French press, right? But Richard seems to like it. So this week in coffee, TWiC. All right, there are a bunch of articles on PDF reading. I think this was also from last week. Tessa had asked, what applications do you use to read PDFs and annotate them and so forth? So Andrew writes, hi, TWiV. I've been listening to the show for a long time, and I love it. It is great while I am working in the lab doing my plaque assays. Keep up the great work. I just wanted to say I second Matt's suggestion from episode 175 to Tessa about the program Papers for organizing references and PDFs. In the new version, you can also use it to cite while you write, kind of like EndNote. I know Tessa wanted an online version, but personally I love Papers. It makes managing references really easy, and it lets you highlight and take notes on articles you've read. If you are a student, you can get a pretty good discount, and they have great customer service. So that's another one. Judy writes, TWiV people! Exclamation point. Evernote is free, but can be used for notes. You can include PDFs, JPEGs, audio, video. Using Skitch, you can mark up papers or PDFs. So, Judy, I use Evernote, but I wasn't able to mark up, and I guess you need a separate application. Skitch, okay. It has extensions that go with Chrome, Firefox, Safari, etc., so you can click and save a URL, article, or web page. You can use it on Macs, iPods, iPads, PCs, Android. The tech support is wonderful. Best of all, it's free. Premium gives extras at $5 a month. I use it all the time across all my platforms. Only limitation is you need to be online, but they are working on that too. And that's from Judy. Your high school science teacher groupie, TWiV? Sure.
Sure.
I kind of like TWiV heads.
Yeah, I like TWiV heads too. Now, also over at the TWiV webpage for that episode, 175, there are some comments on Evernote from Aram. And I won't read them, but he also extols Evernote. He actually put them there because he thought they were, or he or she thought they were too lengthy for the show. But I just want to say...
And that one is addressed, Dear Vinny and the TWiVettes.
I love it. Isn't that great?
Yes.
Yeah, that's good.
We could be a band.
That's very good.
You guys are TWiVettes. Okay. I'm going to let you read this one, Rich.
Okay.
From Neil.
Okay. Neil writes, Dear Vince, Rich, Alan, and Dick, or VRAD. All right. I just listened to TWiV 171. I believe it was Dick who said something along the lines of, quote, wouldn't it be cool if you could label single virions and watch them in real time, end quote. My paraphrasing, I hope I got it right. I wonder what the people at the gym thought when I blurted out, it's been done! Or did I just think of it? Anyway, I'm pretty sure that this has been done for HIV by Tom Hope, Northwestern University, and more viruses by others. Here are some links to related information. So he's got links to images and movies from the Zwang lab at Harvard. And I was looking at that. Let's see. That one is that
It's flu.
flu and polio, right?
That's right.
He's got some labeled flu and labeled polio, and the flu ones are really cool because they get they're labeled with a lipid soluble dye that changes fluorescence when it's diluted. So when the particles are one color as they track into the cells, and then when there's a fusion with a vesicle, it changes. I mean, it's just cool. An article by Dr. Hope that he references, a related blog post, and those are HIV. Anyway, I think the technology is definitely out there. Yes, indeed. He says, keep the great podcasts coming. I learned so much from them. And looking at this, it all of a sudden dawned on me. I mean, we'd heard about this. We'd been discussing this in the past, but it also all of a sudden dawned on me. There is a Vaccinia edition. So I looked up actually, ultimately, I just emailed Brian Ward, who, as a postdoc in Bernie Moss's lab, I think first did live cell imaging of labeled Vaccinia. So he's got a Vaccinia membrane protein that's labeled with GFP, and he's got a bunch of movies. So he's now a professor at University of Rochester. So there's a couple of links on his web page, and I put them here, and there's a whole bunch of links of movies that they made while he was a postdoc in the Moss lab, and I got links there too. So all of these are cool. Yes, you can see single virus particles moving around, and you can even label them in fashions where you can witness their interactions with other cell organelles as well. It's very cool.
How did we let Dixon say that without stopping him?
Right.
Oh, we let him get away with all sorts of things.
Yeah, well, Rich wasn't here on that episode. That's probably it. You would have caught him, right, Rich?
I think so. I don't know. Sometimes I'm kind of asleep at the switch here.
Okay. Alan, would you take the next one, please?
Sure. David writes, Dear TWiV folks, this week one of the founding editors of the Journal of Virology, Lloyd Kozloff, passed away. His daughter, a professor of film here at Vassar College, notified me of his death. Although his name may not be familiar to many, he began his career at a very exciting time as a member of a group of phage biologists associated with Cold Spring Harbor that included many more familiar names like James Watson and Max Delbruck. I can only imagine how thrilling it must have been for a young biologist to work at such an important time in the history of virology and molecular biology. I wrote a brief blog post with links to some of his early papers. Since you've discussed some historical papers on TWiV recently, I thought some listeners might be interested in seeing these as well. JVI will be publishing a more extensive obituary. So very cool to have the blog post. Very sorry to hear about the passing.
Glad that you cleared that up, Alan.
Yes. This blog post is quite good, I thought.
So,
He got his PhD the year I was born. So I figure that makes him on the order of, 25 years older than me? So he lived a good long life.
I didn't know he was a founding editor. Do we know who the others were? It says 3 scientists founded it in 1967.
I do not know.
I don't know that.
Wait a minute. I think it says in his blog. I'd have to look at it, though.
I'm looking here.
Or in his biography.
Anyway, David, of course, is our friend at Vassar, who is a pox virologist, I believe. So
yes.
Thanks for that, David.
Yes.
All right. The next one is from Brenna. Now we're moving into our regularly scheduled email. Brenna writes, Dearest TWiV doctors, how would you approach current and future problems of being research snubbed? About 2 years ago, I was asked to help on a project with another lab. I was struggling to get my own research off the ground, so I had some time. I showed the postdoc how to do plaque assays, but it was obvious he was more inclined to let me do them. So we completed 3 experiments. There's the proverbial so, by the way.
Yes.
we completed 3 experiments before my own research had to take priority over helping him. When things had calmed down, I contacted the postdoc about resuming the studies, but he never contacted me again. At the last experiment, I got the impression that they had a new hypothesis about the experiment and were eager to test it. A few months ago, I see that they have published a paper on that work. Now, no ethics were breached. They never mentioned my work, although to be fair, my work likely gave rise to the studies actually shown in the paper, so there is no ethical issue, just a matter of pride, time, and supplies that were used during their initial studies. I was talking to a peer, and she said that being research snubbed was the reason she required everything in writing prior to working with another lab group. Is that what science has come to? There must be a contractual agreement to protect work and or ideas. It worries me because I love talking and sharing ideas. However, I have already come across one professor stealing my ideas for a grant, being research snubbed, and hearing stories of a researcher stealing ideas from a grant, and obviously scoring the grant low so it wouldn't get funded, and later publishing the research. And I don't even have a PhD yet. What is a young scientist to do? Long-term listener, first-time writer, Brenna. Oh, boy.
Great letter, great issue.
Yeah.
And it's all true, right?
Yep.
Happens all the time.
I thought a lot about this. I have my own spin on this.
Go ahead.
I don't expect to necessarily find agreement from everybody, but my experience with this is to follow the golden rule and do unto others as you would have them do unto you. And what happens? So do the right thing. Be generous. Share everything you got. Share your knowledge. Share your technology, everything else. Never ask for anything in return. Be gracious when it's offered. And sometimes you're going to get screwed. All right? But in the end, nice guys do not finish last. And people will remember you as a generous and good person. And you can have a positive impact on the people around you. You can make a positive difference. There's always going to be jerks out there who are going to rip you off, and there's really not much you can do about it. And if you join that crowd and start, asking for everything in writing and all that stuff, you just make yourself and everybody around you miserable. So that's the way I live. And I've still got food on the table, so it's okay.
I love it. I think it's perfect. I would do the same thing. Well put.
Yeah, and this isn't just, this is not unique to science. This is a phenomenon that occurs everywhere. There are going to be people out there who will take ideas and not give credit, and it's going to happen whatever line of work you're in. You need to develop a philosophy to deal with it, and I am completely in agreement with Rich's approach. Not only because it works out, usually works out well in the end, because people get the idea that, hey, this is a good person to work with, and if you're a good person to work with, you tend to stay employed. But in addition, it's so much simpler, because you don't have to keep track of all of these issues that you have with everybody. And I a while ago, I was talking to somebody about a mutual acquaintance, and they commented, yeah, but he's a bit of an injustice collector. And I thought that term was just so perfect. Because there are people out there who, they end up being very bitter about all of the little injustices. And, there are lots of little injustices that are going to occur to you in life. And you kind of just have to accept that.
Sure.
It's part of the price of admission.
One of the major findings in my PhD thesis got scooped because my advisor, who was was a model for this kind of behavior, very open and free and in exchange with her information, had a conversation with a colleague as I was approaching the end of my work, and that colleague took up with the idea and did a series of experiments very rapidly and published them in a rapid publication journal. And so that was a disappointment. But you know what? I still got my stuff published. I got credit for what I did. Nobody really cared in the end, and it didn't affect my career. And so what? I would do the same thing over again. I would have my advisor do the same thing over again. You know, it used to be, and it still is to some extent, depending on the environment, the tradition that if you knew that somebody else was working on a particular project, you didn't work on it. And in my field, that still happens. I have exchanges with colleagues where we're both interested in the same thing, and we keep in contact with each other to make sure we're not stepping on each other's toes. Now, this doesn't always happen. There's a lot of competition in other fields where people are trying to, run over each other and that kind of stuff. You got to choose your lifestyle, and I choose to just be generous. It's just a lot simpler.
So you're talking about pox virologists, right?
Yeah. So it's.
I mean, you guys play golf together. That's really
right.
It's not the same.
We're very fortunate. And in some fields, it's a lot more competitive. It's a lot different. So,
I think that's the way to go, Brenna. You will find that not everyone feels that way or acts the way we've been talking. Right. But you just have to do, as Rich said, do what you want to do, the way you want other people to treat you. Now, this business about scoring grants low and doing the work, that happens now and then. I think it doesn't happen all that often, and it's really lousy to do that. My philosophy has always been, if you can't think of good things to do, you shouldn't be doing science. If you have to steal from other people, you're useless. So I would never do that, but there are always going to be people that do it. The way the system is set up, you can get away with it, and it's unfortunate, but you have to move beyond it.
But bear in mind that is probably, there are probably more stories about that happening than there are actual real incidents of it. Mm-hmm. So, you said, Brenna, obviously your personal experience, firsthand, but if this is a story that you heard about somebody stealing ideas from a grant and scoring it low, it may or may not have been an actual case of that, because bad grants also get scored low.
Yeah.
And, some ideas are obvious enough that somebody's going to pursue them. So I'm not saying it didn't happen, but you don't necessarily know that all the bad things you hear about did happen.
Mark Twain had a wonderful, typically humorous generalization about this whole thing. He said, always do right. It will gratify some and astonish the rest.
That's great.
That's good.
That's really good. All right. Let's move on to Tina. I believe we are at Rich.
Tina writes, I wonder if you are aware of the most recent study of MS, that's multiple sclerosis, and EBV, that's Epstein-Barr virus. Whereas EBV has not been a trigger suspect for MS since the 1980s, this new study shows that even if the virus is not actively replicating in brain cells of MS patients, it is releasing chemicals that cause an immune system response. I have a cousin with MS. I remember years ago finding out that MS is more common in subtropics compared to other areas. Some studies show it is important where you lived in your childhood and teenage years as to whether you were at risk, no matter where you lived for your adult years. This and other studies prompted the theory that MS is caused by or triggered by a virus. The study in 2006 showed MS patients have an immune system overreaction to EBV. A 2009 study showed, after accessing 7 million blood samples, that those who had no infection of EBV did not get MS. I find this very interesting as I am an ME-CFS patient. As MS has many similarities to ME-CFS. They both have post-exertional malaise or fatigue, much more pronounced in ME-CFS though. They both occur more in women, They both have a relapsing, remitting course for most patients, and they both have occurred in outbreaks. Lesions on the brain can also occur in ME/CFS patients, which, along with the similarity of symptoms, causes some ME/CFS patients to be misdiagnosed with MS. The lesions of ME/CFS are pinpoint, small white spots, but MS lesions are oblong. Ironically, ME/CFS has also been linked with EBV, even originally called chronic Epstein-Barr virus syndrome. Some have noticed increased EBV and other herpes virus titers in ME/CFS patients. It is well established that many, not all, ME/CFS patients develop the disease after a case of mono, such that a study is ongoing following people with mononucleosis to see who develops ME/CFS and possibly why. EBV hides in B cells, which is why the drug rituximab is sometimes used off-label in MS treatments. And in October, a phase 2 double-blind study in Norway found that rituximab, which kills B cells, brings moderate improvement to 2/3 of ME/CFS patients. A few have seemingly been cured. Those physicians theorize that ME/CFS is autoimmune in the brain. Some others have theorized that killing B cells may reduce ME/CFS symptoms because EBV hides there and could possibly be causing a malfunction in the B cells, causing it to make autoantibodies. Kill the B cells and you kill EBV. Could EBV behavior inside the cells be the cause of two autoimmune diseases, depending on what part of the central nervous system is being attacked by autoantibodies? I would love to hear your thoughts on all this. Ron Glazer, PhD, has done studies on CFS and the effect stress has on EBV. Might this be the thing that brings together those who see ME/CFS is triggered by stress, and those who think it is triggered or caused by a virus? And there's this letter is peppered with links to these various studies that she cites.
Well, as many of you know, EBV has been investigated as a cause or a trigger of CFS for a long time, and the results are not conclusive. Although there's new study that she points to is getting quite a bit of press. This is for MS. It's a paper in Neurology. Association of innate immune activation with latent Epstein-Barr virus and active MS lesions. And basically find innate immune responses, production of interferons in active areas of white matter MS lesions, but not in inactive MS lesions. And they would like to correlate that with the presence of EBV latent genomes. It's a small study with small numbers.
Mm-hmm.
So could this apply to CFS? Could it be a trigger? I don't see why not. It certainly isn't present in every patient. A lot of studies have been done to look at the presence of antibodies. The problem here is that, of course, EBV infects over 90% of the population.
Right.
Yeah.
So it's really hard to tease out an etiology in that case, right, with CFS.
Yeah, all of these studies are correlative, and you're talking about multiple factors, and so you don't know. It's always the problem is are you finding EBV because there's a problem, or is EBV causing the problem?
Or is EBV just there because 90% of people have it?
Right. So it's going to be it's not impossible that there's an EBV trigger or a genuine causative association with these things, but it's not proven, and it's going to take a lot of study to work it out.
And we can also say that it's not the definitive or single cause.
Right.
Because 90% of the population doesn't have either
Right.
of these diseases. I mean, that's...
Yeah.
Yeah, when I was thinking about this and looking it over, I was thinking about the paper that you guys did last week on the susceptibility to herpes encephalitis based on genetic background of individuals, and this is going to come up more and more. Is that there are multiple factors involved in a lot of these diseases, involving at least in part pathogens, where a person's genetic makeup is part of the story. So.
Yeah.
The problem is that, genome sequencing is ongoing in CFS patients.
Mm-hmm.
And so you might get some clues, but it's going to be hard because they'll have to look at the entire genome and try and figure out what is causative. In the case of the herpes encephalitis susceptibility, they had known that these innate genes were involved, so that really helped them in looking at the
Right. They didn't do whole genome stuff, right?
No.
They focused on these, right?
Yeah, so I think it's tough, but it has to be done. I agree.
Yeah.
This one study on rituximab, this is a monoclonal antibody that basically reduces the numbers of B cells that you have. So a small study was done in Norway, which found that a number of CFS patients improved after treatment with this antibody. But the improvement took a couple of months to kick in. So the authors say it's not likely that you're getting rid of a virus in these B cells because that would have happened right away. Mm-hmm. So they think that it's immunosuppressing and causing autoimmunity type reactions to regress, which would take a couple of months. So I'm not sure it's consistent with EBV being there.
Right.
But
I didn't see
It's also a very small study. So
yeah, it's really small.
Right. I didn't see, EBV is in B cells. If you get rid of B cells, maybe it has some effect on these patients, but that doesn't necessarily add up to say that EBV is causing this.
Yeah.
Okay.
Right.
I mean, it could exacerbate it, but I think there's something else, whether it's another agent or a genetic lesion or both. It just needs to be worked out, and it's complicated.
Yeah, and I, as I've said before, I will be surprised if ME/CFS is not a multifactorial disease.
Mm-hmm.
Right.
It's just the way the epidemiology goes on that and what's known about it. It seems like there shouldn't be people saying, "Oh, it's got to be caused by X," or "It can't possibly be caused by Y." Nobody's excluded anything.
Do you know if there's such a thing as familial ME/CFS? Is there any sort of evidence for a genetic component? Because I was thinking if you just go doing genome sequencing, you're going to probably come up with a mess. Usually, when you get somewhere with that, yeah, I don't think so.
Part of the problem is probably that it has only. I mean, it wasn't really. If you go back 50 years, nobody was diagnosed with it.
Right. Yeah. Okay. So you're not going to have a big family tree, right?
Yeah.
Okay. Good point. All right.
So I'm looking at a site. It says there, it is seen in members of the same family, ME/CFS.
Okay.
Which is believed to be a familial link, but nobody's proven it yet.
Okay.
Right, which could also be an environmental link.
Yeah, sure. Absolutely.
Right.
All right. I was going to say one more thing about that. Oh, a couple of weeks ago, with Kathy Spindler, we talked about a paper where two viruses cause disease in an organ where one or the other doesn't by itself. Mm-hmm. So this could be something like that where it's hard to tease that out.
Yeah.
Keep our eyes open. All right. Alan, I think you're up next.
Yep. Greg writes, "Hi guys, great podcast. I'm a PhD student at McGill and I work on HIV, but in more of a biochemical context than a virological one. Your podcast is great at rounding out my knowledge of the virus I work on, and of course many other viruses as well. I'm sure this paper is on your radar as it comes out of David Baltimore's lab, but I think gene therapy is an amazing technology, and this seems like a perfect application in order to cure a difficult virus, HIV." And he links to the vectored immunoprophylaxis paper that we talked about in the ferrets runny noses episode.
Yep.
So yes, that was on our radar, and we're just finally catching up on our mail from before that episode. But thank you very much.
All right, the next one is from John. Dear TWiVers, I loved your Concerto in B episode at 161. Your discussion left me with a bunch of questions regarding B cells and antibodies. As I was listening to the discussion of affinity maturation, I kept trying to think of how this process interacts with existing memory B cells, and in particular how it interacts with original antigenic sin. What happens when you already have a good population of memory B cells to an antigen? Do you get new germinal centers forming and the affinity maturation process going on again? And if so, Does it start with the B cells that have already been through that process, or does it start with new immature B cells that responded to the soluble antigen? Does that depend on how much new antigen is available? If you have an effective antibody response, does that keep this process from restarting? All right, so let me stop there and tell you that I sent these questions to Gabrielle Victoria, who was on that episode with us, and he answered each one. So let me answer each question as I read them. So this first batch, Gabrielle responds, let's see, I'm so confused. Yes,
you should start with his preamble.
Yeah, the preamble is good.
Yeah, I was going to read that later, but okay, I'll read it. Please congratulate your listener. These are all excellent questions. Unfortunately, I don't think we know nearly as much about these things as we should. Vaccine development has historically been mostly empirical, and regimes are usually based on what works best rather than on immunological considerations. Most vaccines were actually developed by microbiologists.
As opposed to immunologists.
Interesting. Part of the difficulty in addressing these issues is technical. It would be very difficult and costly to serially determine the affinities and specificities of B cells responding to antigen during consecutive immunizations of the same individual or animal. All right, so this first batch of questions, he responds, the original antigenic sin phenomenon is most likely related to the rapid elimination of antigen by existing antibody or by antibody produced soon after the second challenge by existing memory cells. The novel... sorry.
I'm thinking we ought to redefine original antigenic sin for those who are not tuned into this. So I'm not sure I can do it, but if you have mounted an immune response to, say, one strain of flu, and you're subsequently infected with another somewhat immunologically overlapping but distinct strain of flu, you can have an immune response that is specifically to the original strain, not to the novel strain, okay,
right.
Which is a little weird, okay, and that's called original antigenic sin.
That's right. Okay. As to new germinal centers, emerging data suggests that some memory cells, those that haven't switched their antibody isotype, can make it back into GCs upon second exposure. I would expect that a secondary GC would therefore contain a mixture of B cells derived from naive and memory precursors. Many factors could influence the composition of this putative mixture, including competition between B cell clones for limiting amounts of antigen or T cell help. All right, so that was his first part. Then,
so he's thinking that original antigenic sin really has to do with the fact that since the second antigen is related, it gets wiped out before you can really make an immune response to that. Mm-hmm. And he's also saying that isotype switching, so going from IgM to like IgG or something, is really a commitment step in B cell development, as I see it. So once you do that, you're not going to go back and get retrained.
Right.
Right?
That's it. Right. All right, so the John continues with his next question, which I've lost. Here we go. Do some mutations in the affinity maturation process close off the possibility of mutating back in some way that would be helpful later? Or is all the raw material still around in a functional B cell after affinity maturation so that you can get from a good antibody to the 2005 flu strain to a good antibody for the one you get in 2007? It seems like there must be some impact of the existing stock of memory B cells on future affinity maturation, or you wouldn't observe original antigenic sin. And it seems like it should work the same way when there's antibody-dependent entry of cells as with dengue. Is this right? So Gabrielle replies, I think this is mostly a matter of the extent to which the existing memory cells recognize the newcomer variant. If it is enough to trigger an original antigenic sin type phenomenon, you would probably not generate new germinal centers, in which case you would get no further affinity maturation. However, naive B cells are constantly being turned over, dying and being regenerated in the bone marrow. Therefore, the likelihood of a hole in the repertoire in 27 arising due to all flu-specific B cells having gone the 2005 way is negligible. But theoretically, it is possible that an unswitched 2005 memory cell could re-enter a GC in response to 2007 viruses. And in this case, it is conceivable that some cells could be impaired by 2005 specific mutations, but these would be unlikely to last in the highly competitive environment of the GC. It's getting pretty technical, folks.
These are great questions. These are great questions. Really, I mean, he's got an efficiency question here. You know, it seems a waste to do all that affinity maturation and then not be able to capitalize on that when a related antigen comes around. So can you take those memory cells and go back and, you know, they've already got a bachelor's degree. Can you give them a PhD, right?
Right. Or for a computer analogy, why wouldn't you save your old versions?
Right.
Yep. Yeah. All right, is this process the reason why some vaccines require 3 shots to get protection? I recall getting the Hep B vaccine many years ago in 3 doses. The second shot a month after the first, the third 6 months later. Or is this more a matter of getting enough antibodies rather than getting higher affinity antibodies? The 3 shots of vaccine, this is Gabriel's answer, probably have more to do with attaining high titers of circulating antibody in blood than with improving affinity. Serum antibody affinity already increases quite dramatically, up to 10,000-fold within the first immunization, although serum antibody titers, a composite measure of affinity and amount of antibody, only really go sky high after 1 or 2 booster shots. The idea is that whereas the first contact with antigen generates germinal centers and affinity-matured memory cells, the booster doses reap the profits of this germinal center reaction by triggering the ensuing memory cells to proliferate vigorously and differentiate into large numbers of antibody-secreting cells within a few days. What the role in vaccination is of GCs emerging during booster doses is not clear, at least as far as I know, but could conceivably involve the recycling of unswitched memory B cells back into GCs for further affinity maturation. That makes sense?
Mm-hmm.
Yep.
So there is one exception here, the polio vaccine, which has 3 serotypes in it. So this is the orally administered infectious version. That's given 3 times because each time you give it, one serotype usually wins in your gut. And so it replicates, you get a good immune response. Then you go back again, one of the other 2 replicates, and then the third time, the third one does. So that's different from what Gabrielle is explaining here.
But that's unique to that.
It's unique to polio,
particular
I
vaccine.
believe.
So up to this, as I read this, up to this point in this letter, it's mostly about can you further educate memory B cells by recycling them through old or newly established germinal centers.
Exactly.
Excuse me?
Exactly.
Can you teach an old B cell new tricks?
Right. And it sounds like Gabriel's saying,
Probably not, right?
probably not, or at least it's, certainly not known with any certainty, and he seems a little skeptical that this is going to be a very common phenomenon.
Right.
Am I reading that right?
Yeah, I would say that's correct, right. Finally, listening to the description of the germinal centers, it seems like they should run out of antigen since they're using it up each time a B cell tries to take up and present antigen to T cells. Is there some mechanism to recycle the antigen? Is the supply of antigen the limiting factor in this process, or is there plenty? Maybe my intuitions at this scale are all wrong.
I had the same question after that podcast.
And Gabrielle responds, this is a good question. Antigen appears not to be limiting since it can be detected on follicular dendritic cells many months after the GC reaction has faded, though how available this antigen is at this point is not clear. Many hypotheses exist for why the GC response ends. These include competition between B cells and circulating antibody, which would sequester the antigen available on FDCs, and exhaustion of GC T cells, perhaps through their regulation by GC-resident regulatory T cell populations. I tend to favor the latter, but this is still a topic of fervent investigation.
Yeah, I remember that thing about follicular dendritic cells hanging on to antigen and being there to present them.
Yes, I remember that
during
too.
the maturation, and apparently indefinitely, and that to me is magical. because you're not talking about a lot of antigen. But it hangs around for a long time, and I guess that's a mystery.
So we all have
these
a history.
are amino acid sequences.
Yeah.
Yeah.
How do you hang on to that?
So we all have a history of all these infections in our
Well,
GCs.
apparently, eventually the GC goes away, right? And I would assume at that point,
that's
it's
right.
over. Okay, but it's there for apparently as long as you need it, and the response doesn't end because you run out of antigen. It ends for other reasons. Okay, the antigen is not the limiting factor.
All right, John writes up in closing. Anyway, thanks for your wonderful podcast and for answering my amateur immunology questions. Apparently they're not.
All that amateur.
And we thank Gabrielle for answering them because I don't think any of us could have.
Yeah, and I need to go back and listen to that episode again because that was just a wonderful episode. It was great stuff.
All right, who is next? I've forgotten.
I think it's me.
All right.
Is that right, Alan?
I think it's you. Yes, Vincent, then you, then me.
Okay. We're going
We're going clockwise.
clockwise.
VRA. That's right, VRA. We're just missing D.
John writes, hello. Attached are my two questions. I found your podcast even before the death of my sister during the 2009 H1N1 pandemic of 09. She died 14th of October 2009 from bilateral pneumonia as a result of a confirmed case of H1N1.
I'm sorry.
Yes. Yeah. First order of business, our condolences. I received both the seasonal and H1N1 vaccines, as I'm not stupid and not influenced by the celebrities or former Playboy bunnies, ref Jenny McCarthy, name intentionally lowercase out of disrespect. I prefer my medical advice from a doctor. Imagine that. I'm sure I'll have more questions as I make it through the episodes. Respectfully yours, John. And he left us 2 recorded questions.
All right, I'll play these each separately. I don't know if you guys will hear this. That means Rich and Alan, but I presume you've listened. So
I have listened.
When I am done, we will. So the first one is about virulence.
Hello, TWiV Meisters. This is John from California. I'm just catching up from the 2009 podcasts during the H1N1 outbreak. Earlier, Dr. Racaniello stated that influenza will become non-pathogenic in temperatures over 75 degrees Fahrenheit, while our body temperatures are nearly 100 degrees Fahrenheit. I was wondering why they don't become non-virulent or non-pathogenic in our bodies. Thanks for your time.
You guys hear that?
No.
Yes, I did.
Oh, you heard it? I didn't hear it.
All right, this is the one.
I had played them a while ago, but I forget what they were.
So this one, he's citing me in an older podcast as saying that flu doesn't become pathogenic over 75 degrees Fahrenheit, and he wants to know, our body temperature is 98.6, so what's that about? So I cannot remember what he's talking about. I don't remember saying that. It wouldn't be correct that it wouldn't be pathogenic. I'm trying to think of what he might be thinking about.
So this was probably in relation to the flu season discussion. Why does it correlate with cold weather?
Yeah. So we know that transmission is more effective in conditions of cold and low humidity. I don't know where the 75 degrees would have come. That might have been from discussing that curve. So we were thinking that it's inactivated at high humidity or high temperatures, I should say. So that may be what you're thinking of.
Right, but those are high outdoor temperatures.
If you could give me a specific podcast number, I'll go back and listen, John, okay, to see if that's it. So that's what we think is going on. All right, so that was one. The other one has to do with the names of viruses, and I'll play that now.
Hello, this is once again John from California. I have a question. I heard on, I believe it was the Naked Scientists, quite some time ago, probably a year ago, that that we use the word viruses because it came from Latin, I believe, and they didn't have a plural for the word virus. But, however, in Latin, the plural would have been virii. So my question is, why didn't they use the word virii as a plural, or just virus by itself as a plural? And I ask this because doctors and virologists in each language are going to use their language's version of pluralizing the word virus. This is confusing, and it just bugs me to no end because I'm such a perfectionist. Thanks for your time. Bye.
So, Rich, that was,
It's about what the plural should be.
Yeah, so virus is Latin, and the plural is viri. So why do we use viruses?
It's because we're, idiots. We
I asked Dixon. He said because we don't speak Latin.
That's right. Exactly.
Although virus is from
What do we know?
Latin is virus is poison or slimy liquid. I remember.
Right.
So viri is not a word that would persist in the English language. What do you think, Alan?
Well, looking this up, it seems to come down to a question of Latin and English grammar. So virus was originally a mass noun, which would not pluralize in the same way as ordinary nouns. And so viri may not be the correct plural for it. And so viruses became the standard that we use.
Whoa, that's heavy duty.
Yeah, I looked it up. I thought, okay, I'll just to take the Latin scholar's word for that.
I kind of like viri myself.
Some people use it jokingly.
Yes, right.
But none of us do. So if anyone has some insight, let us know.
Yep.
All right, Alan, you're up with Raihan.
Raihan writes, hello, insert cool way of addressing you guys. In one of your previous TWiVs, you guys mentioned about how after the H1N1 pandemic, the prevalence of flu infections dropped, suggesting a boost in herd immunity. If you don't mind, I would like to chirp in on this discussion. I'm a grad student studying influenza here in Singapore. In my confirmation presentation, which was at the end of the H1N1 scare, I presented the following chart taken from the Ministry of Health, Singapore's website. And he sends along a chart showing weekly prevalence of different flu strains.
Right, both H1N1 and seasonal and
flu B. So this seems to show he describes the pink bar, pandemic H1N1, shows a decrease in size over time corresponding to the end of the pandemic. But what is interesting is that while pandemic H1N1 dwindles down, the incidence of seasonal flu A strains and flu B increases. I'm curious as to whether or not the same trend is observed in the U.S. You guys seem to suggest that after the pandemic, the incidences of seasonal flu and flu B were not as prevalent. This is obviously not the case here in Singapore. Let's go ahead and take that one first, I think.
Okay,
and my first thought is Singapore, being in the tropics, is going to have a different flu dynamic than temperate climates, I would expect. So, Vincent, you just popped in the link for the CDC.
Yeah, so this is a link to the summary of the pandemic season here in the U.S. It's 2009, week 16 through week 39 of 2010. So you can see week 20 you have an increase. That was like March, April of the new H1N1. B at the time was very low, and that was extinguished as soon as the new pandemic strain emerged. That went down over the summer. Then towards the end of the summer, it peaked at week 45 of 29. And there was no more B activity after March or April in that year. So it was quite different from what you see in Singapore. And we have no idea why. When B is usually extinguished can be by competition with A strains. And so that could have happened here in the U.S. And as the pandemic strain diminished in Singapore, it might have been replaced by the B strain there. So that could explain it. But people really don't understand the dynamics of these two viruses. That's my 2 cents anyway. Right now we have hardly any B activity in the U.S. Of course, flu activity is very low, but B is way, way below the A's.
Yeah. Okay. On a separate yet related topic, I have to express my dismay in your podcast for not highlighting other types of influenza. When you guys talk about flu, you exclusively talk about flu A. This frustrates me as I am studying influenza B. I have only noticed influenza B being mentioned about 2 to 3, and from this point on, he capitalizes all
That's B. Yeah, this is terrific.
About 2 to 3 times in the course of your podcast, and it was always in passing. The only discussion about flu was by Peter Palese, where he suggested that flu B could be potentially eradicated due to its lack of an animal reservoir. Please don't get me wrong, you guys are doing a brilliant job with the podcast, but flu B is my baby. I would love to hear your insights about flu B as well.
May I chime in here before you go on?
Yes.
So, Raihan, I did my thesis on flu B.
All right.
That's a long time ago, and I haven't talked about it since then. But
And you still have food on the table, too.
I do. So we could. It's actually an interesting virus. We'll dedicate some time in a future podcast to talk about flu B. You're right. It gets disrespected. I don't know why.
Okay, so we'll respect it.
We will.
That's right. Well, flu A tends to push it out, so we pay attention to the one that comes up a little bit more often.
Also, we should point out that there's very little antigenic variation with flu B, and hence it's less of a disease maker, if you will.
But is pretty consistently in the vaccine, right?
It is always in the vaccine because it does cause morbidity, but it's much less than A because, it gets wiped out by an antibody response quite quickly.
Right.
And why is there less antigenic variation? I think this is a mystery, right?
Yeah, this is a mystery. I mean, one interesting aspect is there is no animal reservoir for flu B.
Mm-hmm. But that wouldn't necessarily impact on drift.
Wouldn't impact drift, but it would, of course, prevent pandemic strains from emerging.
Right.
But the drift, we don't know. That's a good question. We'll look into it and do a show on it.
Okay. Yeah. Maybe we can get a flu B person on.
To be or not to be would be the title.
Yes, we've already got some titles.
Yeah.
All right, finally, my take about the are viruses alive or not debate. I've been thinking about this for quite some time, and I totally agree that this is not exactly a biological problem as much as a philosophical one. As early as I can remember, amongst the first few things taught in biology is that the cell is the basic unit of life. I think this is quite an agreed-upon definition, and no one would disagree with it. Most of us would see this in biology textbooks in all levels. Therefore, since viruses are not classified as cells, it would be impossible to define them as alive as much as organelles are not alive. Hope my email did not bore or bother you guys. Just take my earlier complaint about flu B as the rantings of a PhD student trying to come to terms with his lack of publishable data.
Just keep
nice
at it.
letter.
Yes.
I like his take on viruses.
Sure.
Yeah.
I like them all. All right, the next one is from Marcy, who writes, Dear Twivers, I am listening to episode 164, 6 Steps Forward, 4 Back, and as you were mentioning the fact that you wished you could reach everyone, it occurred to me that the podcast title, This Week in Virology, is a little intimidating to people who may only have a high school knowledge of science. I am not suggesting that you change the name of the main show, but maybe you could do an occasional single topic, explain it to the layman show, where there are hot topics like the HPV vaccine, the H1N1 spread, or the H5N1 publication controversy. These could be short, 15-minute shows that target a broader audience than your regular shows. The name would need to be something general, so as to cast a wide net. All of immunology, microbiology, you certainly have sufficient connections that you can recruit experts to do bacterial or fungal topics, if not broader. It would also need to be non-threatening to the average person. Think Idiot's Guide to. So it would need to avoid words like virology and microbiology. Maybe something like hot topics in health and disease. And that's from Marcy in Pittsburgh, where it is currently 50 degrees Fahrenheit and overcast.
At least when she wrote it, which was a while ago.
Could have
Probably.
been last summer. Who knows?
We are behind on email, yes.
I think it's a good idea. So do you guys think that virology is intimidating to a lot of people? It's interesting.
I run into people who, they do a double take when I say virology, and they have to think. I usually have to follow up with saying that means I study viruses, right? Because it doesn't it doesn't resonate immediately.
But everyone knows what a computer virus is, right?
True, but the ology, virology, throws some people a curve. We don't talk about computer virology. And she's right. When I tell people, ask me what I do, and I'm a professor. Okay, right. What do you profess? I say microbiology, and they go, ooh, like that's a big deal or something. Okay. So it is. I guess those terms are a little intimidating. I hate to, in a way, sort of.
Dumb it down?
And I hate to sort of perpetuate the notion that those words are intimidating because, in fact, once you're familiar with the idea, it's not intimidating, okay? It's not a big deal. It's the study of microbes. It's the study of viruses. It's not a big deal. But she's got a point. If you're chasing people away with a couple of words, then maybe you could do something about that and get them sucked in and then slam them with virology.
I think she's right, but it involves probably a separate show, which it's tough to do at this point. But
because people who are intimidated by the word virology are probably going to be intimidated by a 90-minute podcast too.
Yeah. So Daniel and Natalia, remember that when you do your podcast in Spanish. Maybe you should leave virology out of the title. I don't know. Depends who you're targeting. All right, the next one is from Sven Urban. Who just read the last one? I did,
think
right?
you did.
So it's Rich.
Right. Good, I'm glad I get to do this. Sven Urban writes, Ave Magi, I'm screwing it up. Magi virorum. Let me do that again. Would you say magi?
Magi, I think.
Magi, Ave Magi virorum. He says, parentheses, that ought to put me pretty high on the list of creative/obscure greetings, exclamation point. Yes, indeed.
I think you may also have just raised Cthulhu.
Yeah, it's pretty funny. That's a Lovecraft allusion, right?
Yes.
I'm gonna read his P.S. first. P.S. The greeting supposedly means "Hail ye, magis of viruses."
Mages. You're certainly not a video gamer.
No, I'm not. Mages of viruses. Well, I had to look up magi, okay, and find out because I always think of with my Western background, I always think of the three kings and that kind of stuff, and I figured that it's kings. No, it's magicians, right?
Is it?
Yeah.
Magi so that's what a mage is, a magician?
Well, yes. I don't know if that's what a mage is, but it's what magi is. Okay, if you go way back, it's an ancient word, and it's basically magicians.
Yeah.
So I like that. Magicians of viruses, at any rate. Hail ye mages of viruses. Kudos to internet if I'm right. Shame on me if I'm wrong. Well, yes, that's very high on the list of creative obscure greetings. I like that. I have just enjoyed episode 169 where you all discussed the question from Sophie on how to read scientific papers. I fully enjoyed and appreciated your learned views, all of them very valid for you as scientists. However, not all readers of scientific papers need be or become scientists, so perhaps you might let me add one layman's perspective. Just to give you the general picture, I am a software quality professional with a 30-year-old MSc in computer science. Out of general curiosity and for personal entertainment, I occasionally read science papers from other disciplines, mainly from the section Evolutionary Biology in PLOS One. Many of the intricate details in those papers, whether they be on primatology, paleontology, ecology, or particularly biochemistry, are usually beyond my grasp and knowledge. That's fair. Scientific papers aren't written for laymen, and Alan's way of describing a paper as a highly compressed packet of information for transmission over distance was very instructive. What I look for and hope to find is something reasonably digestible at the beginning and end. As Dixon pointed out, the pictures and graphs are very important, as they may aid or sometimes hinder if badly designed, the intuitive understanding of the data, and here too good handiwork might aid in my understanding. With well worked out introductions, discussions, and illustrations, I, the layman, should be able to grasp the general problem, question, hypothesis, as well as the general result, conclusion, implications. And that's my main point. On some level, a scientific paper ought to be accessible to the non-initiated. If it's not, the paper and science is in some sense poorly or obscurely presented. On the other hand, if science papers were always graspable by the populace, Alan would be out of business pretty soon. Of course, as a layman not understanding the details, I have no ways of assessing the validity of the results presented, but that is of less concern as I am not using the information gleaned for anything else than maintaining my image as an incurable intellectual around the coffee pot at work. Thanks for your continuing effort, Sven Urban. Good point. You know, I really like this. The notion that a scientific paper, no matter how specialized, ought to have maybe enough in it so that a person not specialized could look at it and at least get some idea what's going on.
Yes, and at least a couple of journals that I'm aware of do kind of nudge things a little bit in that direction. PLOS has these author summaries.
Yeah, I was just looking at that.
So there's the abstract, which is the super condensed version of the paper.
Yeah.
And then there's an author summary that is supposed to be just kind of the author talking to everybody and saying, okay, everybody, this is what this is about.
Yeah.
The other journal that comes to mind for this is actually the CDC's publication, MMWR, Morbidity and Mortality Weekly Reports, where they'll do a detailed report, and then after it, they'll have an editorial comment. And the editorial comment will be a few paragraphs kind of putting the whole thing into context. And so there's some effort in that direction, but I absolutely agree that scientists should be encouraged and probably even taught to start their introduction in a way that somebody who has one college science course would be able to understand, and then you can get into the hardcore stuff, and then try and wrap up your discussion, in the way that same one college course experience person can get it.
Yeah, I agree. I try and do that when I write a paper to make it clear for almost everyone, but you end up getting into abbreviations that are pretty crippling, I think.
Yes.
But it's a good thing to do, at least in theory, to try and make it a little bit more accessible, because you never know who's going to read it, especially with the popularization that's going on, right?
Yeah.
I mean, all the people that look at our links, right, alone.
Right.
All right, the next one is from Coleman. I believe we're at Alan now. Yes.
I think it's my turn. Colm writes, Sirs, I've been listening for a while and finally have enough disjointed comments to merit hitting send, I hope. First, you frequently make references to temperature conversion. Born and raised in the United States, I have a poor understanding of Celsius as it relates to real life. I know my incubators are at 37, and that's toasty, and the lab is in the low 20s, and that's room temperature. I wonder if you know of a weather application for iOS or Android that displays Celsius and Fahrenheit side by side for building those associations. I think it would be an amazing feature. Secondly, I'm not a Redditor, but I was recently made aware of their Ask Me Anything threads where experts, insiders field questions from the community in long-running dedicated threads. For example, George Pelikanos, a writer and producer for The Wire and Treem, recently answered dozens of questions in such a thread. It would undeniably be a massive time sink, but maybe a useful way to disseminate virology to the public. That's the site Reddit. Third, and finally, you mentioned in this week's epidemiology TWiV that BSL-4 facilities seem to be predominantly government-run affairs. That is generally true, but I'm aware of at least one privately operated US BSL-4 in San Antonio at the Texas Biomedical Research Institute. The more you know.
I don't know if that app, does anyone?
No,
I'm sure
neither do I.
It must exist, though, right?
No, I know of, there are a lot of unit conversion apps, certainly.
Yeah, but you got to play with them. He's talking about something where the Celsius and Fahrenheit show up side by side all the time to help make the associations. I think that's a brilliant idea.
Well, I'm sorry if this is hopelessly old school, but you could just you could print out a piece of paper.
Alan,
side by
Come
side.
what are you going to do? Carry it around with you?
You can make it a card.
Actually, you can make a PDF and stick it on your phone in, Docs to
There
go.
you go. There you go.
Yeah.
The old guy comes up
Right.
technologically advanced. Cool. All right, I'm afraid we have to do just one more.
Oh, no.
Right, it's already an hour 45 minutes.
Are you kidding me?
No.
Jeez, time flies when you're having fun.
I didn't think we'd get through all these. It's amazing. But anyway, one more, and this is from... I've lost my place. This is from Diane. Because I have CFS, I learned about you and your various podcasts during all the XMRV hoopla. After many years of struggling, I have been able to teach part-time for several years now. I teach biology at a junior college, both a majors and a non-majors course, and I thought you would appreciate something that happened early on this semester. I always start my classes with what I call bioliteracy topics. I will briefly introduce something in the news that is both interesting, I hope, and usually controversial. So, of course, I chose as one topic the H5N1 controversy. At the time, my understanding was that the mortality rate was near 60%. Meanwhile, I had decided to listen to all the current TWiVs, TWiPs, and TWIMs, and to slowly go back and listen to all of the old episodes. So, of course, you know that I had to correct myself with my students the very next class. It actually helped me to make a point that I love to make, that science is not merely a collection of facts to memorize. It is a process. Those who undertake science must maneuver through this process, and the public needs to understand that at any given time, the information they have may not be the final word. And in fact, what they are hearing in the media may be misleading or simply incorrect. I allow my students to earn some bonus points by responding to the bioliteracy topics on exam days, and several chose to write about the H5N1 controversy. I'm pleased I was able to provide them with information beyond the headlines. Thank you for that. And that is just beautifully put.
Absolutely. Diane, keep teaching. Boy, we need teachers like you. This is great.
It is a process, absolutely.
Yeah.
And that's what I tell my students at the beginning of every semester of virology. It's not memorizing facts. It's the process of getting them. That's what science is about. And I love this idea that what you're hearing is going to change. That's the way science is. People have a hard time with that, don't they?
Yeah.
And, you guys did a show a while ago that I wasn't on where this topic again came up of how to communicate or teach science. How much science should we try to teach to the public, or how much can you teach? And to me, once again, it's not the facts. The important thing is to teach the method and that it is a process. If we could get that much across, then I think you've communicated a lot. So she's doing a great service with this.
Yeah, and the information is going to change, but it will change in ways that converge on something that will ultimately be true.
Right.
That's how we do this.
Right.
So it's constant course corrections.
Kind of like sailing, huh?
Yes.
Kind of like sailing.
Well, you always said that, Rich, right?
Yeah. Did I say that?
Yeah, sure.
I'll take credit for that.
You do now.
Let's do some picks of the week. Alan, what do you have?
I have a cool site that I just stumbled on recently called Chronozoom. So this is the history of the universe on a web page. And it took me a couple of minutes. The controls on it are a little bit odd. But if you zoom around on this page, so it's at the very top, you've got a timeline that's marked in billions of years ago, and it starts 13.5 billion years ago and goes all the way up to the present over on the right. And if you put your mouse along that line, a bunch of things drop down where you can go to different thresholds. The origins of the universe, the origins of chemical complexity, origins of the Earth, origins of humans. And if you pick one of these thresholds, then you get a little link that comes out, and it gives you a summary. And if you go to one, you get a link to a video, which is scientists talking about these various things. So you'll get a cosmologist talking about the origins of the universe. You'll get an evolutionary biologist talking about the origins of life. So it's this whole package of discussions about how these events took place.
It's pretty neat. Your mouse.
I cruised through some of this. I cruised through some of this and listened to several of these. I agree with you. The navigation is a little awkward to start with, but you get used to it, and it's terrific. I would like to sit down and spend a long time with this. This one guy, David Christian. It looks like they've culled videos and content from a number of different places.
I think so.
Together in this, and this guy David Christian is fascinating. He's conversant on. I mean, he shows up throughout this, from the Big Bang all the way through human evolution. Okay, and that's from some sort of series or something. And he's a very good speaker and a fascinating guy. But this is a great site. I really like.
It's a great way to aggregate information. And your mouse wheel also zooms in and out. It's very cool.
Yeah. Oh, I didn't see that.
You could do this for the cell, you know.
Ooh, it does. Holy cow, I didn't see that.
Right? A cell. This would be perfect for a cell.
Uh-huh.
You start with a cell, then you could zoom in on different parts and get lectures and information from different people. You could even have a virus-infected cell.
Yes.
That would be nice. That's cool.
Very cool.
Excellent.
Very nice.
Rich, what do you have?
Okay, I have something that Kathy Spindler sent me. And we were having a little email exchange, and this kind of just came up. And it is a site that's a sort of a sublink from the Center for Communicating Science at Stony Brook University, which is a center that Alan Alda has a large part in creating or running. And just to give an introduction to this, he's got this site is The Flame Challenge. And he's issued a challenge to scientists to submit explanations for what a flame is that could be easily understood by an 11-year-old. And I've got a couple of other links here, a science editorial and a New York Times story that are sort of background on this. So Alan Alda has been doing science narration and science promotion to the lay public for a long time in a really nice sort of way. And he has an interest in communicating science to the public. And this is sort of one of his efforts. I asked my father this question when I was a kid. What is fire? What is a flame? He was a chemist. I figured he would know. And I think he told me it was glowing carbon. And that was not satisfactory at all. And I still can't answer the question. I mean, I got some ideas. I want to see what this comes up with. At any rate, both this flame challenge looked interesting, and I was fascinated to see this Center for Communication of Science. Do you know about this, Alan?
I am certainly aware of it, and Alan Alda, of course, is huge in science communication. He's been doing this for years. But I had not come across this flame challenge, but I think it's brilliant. Mm-hmm. I think it's a wonderful idea.
And so we should follow this and see what comes out of it. And the Center for Communication of Science is there to educate scientists about how to communicate with the lay public, because that's part of what he's doing here. And the Flame Challenge is an obvious example of that. You know what we ought to do? I was thinking of this earlier because we had a lot of letters in this show that were relevant to this. Let's get Alan Alda on TWiV. What do you suppose are the chances?
He should, because we're all about doing this.
Yeah.
But it's whether he thinks we're worthy or not. Who knows? I think he should even be listening if he's interested in teaching the public
maybe
science.
he doesn't know. Send him a link.
All right. I don't know if you can get to him. That's the problem. You get to an assistant or something. His people.
His people?
I think he would be great to have on and talk about viruses and educating stuff.
Yeah.
I think it'd be great. Sure. We'll try it. Nice. Yeah, I had seen that. And there's a New York Times article where it starts out with this. When he was a kid, he asked his teacher, "What is a flame?" And the teacher said, "It's combustion," and he didn't like that, right? And then at the end of the article, the writer said, "So what's a flame?" And he says, "Combustion." Of course.
Yeah, both the Science editorial and the New York Times article are nice. They're a good background.
All right, my pick is an article, or an opinion, I should say, in The Scientist magazine called "Opinion: Academic Publishing is Broken. The current system by which academics publish their scientific discoveries is a massive waste of money." So this follows on a variety of discussions we have had about publishing, open access, and otherwise being behind a paywall, and most recently, the law that was attempted to be passed to block open access publication by the NIH we talked about a while ago by Elsevier. And this basically goes through a very interesting argument on why the way we do it now is bad and it's broken. I love it. I really like it a lot. But read it. It's really good and it won't take you more than 10 minutes. Academic publishing is broken. I've thought this for a while.
Yeah,
myself.
it's a good article. It really kind of lays things out.
Yeah, I thought this was brilliantly written, and he lays it out very simply.
Well
very
done.
nice.
All right, we have a couple of listener picks of the week from Joel. Hi, TWiV. The CDC's EIS program has been mentioned on several TWiV episodes. I would like to nominate Tyler Sharp as a potential guest. I worked with Tyler in Michelle Hardy's lab in the summer of 23. I had never met someone so passionate about virology as Tyler. One of my most vivid memories of him is how he carried your Principles of Virology textbook around with him all summer. He went on to earn a PhD in Mary Estes Lab and is now a lieutenant in the EIS. Tyler worked recently in the Marshall Islands effort to control dengue. He wrote about this experience in the CDC's Public Health Matters blog. Even if it doesn't work out to have Tyler as a guest, perhaps his 2-part blog post could be a listener's pick of the week. Yes, that's why I picked it. Now, if he carries my textbook around, he should be a guest on TWiV. But I couldn't find an email anywhere for Tyler.
Is that right?
Yes,
Not
I
even
tried.
on his blog, huh?
On his blog, on his papers, I couldn't find it. So, Joel, if you can get an email, I'll invite him.
Great. The blog is terrific.
Yeah, it's very nice. All right, the second is from Sven Urban, who we had an email from earlier. Searching my bookshelves for a listener pick of the week, I found something that at least distantly tags in with the main theme of the episode. That's the episode on reading a paper, I suppose. I suggest 2 highly accessible books on how we tend to overly rely on noisy, possibly meaningless data and how we tend to extrapolate trends ad infinitum and how we cannot disprove the odd/unknown/unobserved phenomenon. Fooled by Randomness and The Black Swan, both by Nassim Nicholas Taleb. Okay.
By the way, I am sending you. I just searched up an email address for Tyler Sharp and also a phone number.
You got it?
Mm-hmm.
How come I spent 20 minutes and I couldn't get it? Never mind. You don't have to answer that.
Finding contact information for people is
That's
a
your
big part of
specialty.
my job.
Very good. Thank you.
Okay, so find Alan Alda.
Yeah, find Alan Alda, not his people.
That's good. Well, you're right. That would be finding his people. It'll be very easy to contact his people through the Center for Communication. Just go through the front door.
Well, if I wrote to them on my Columbia email, maybe it would help, right?
Definitely. Send it from your university email and tell them you're the host of a podcast with over 10,000 listeners, and
Yeah, and wait till this one is live, so you can send them a link to this one where we actually hype him.
Yeah.
Yeah.
And that will do it for TWiV 176. You can find us, of course, at twiv.tv on iTunes or the Zune Marketplace. And if you haven't done so already and you like us, go over on iTunes and leave a review. It helps us to stay prominent and have more people discover us. By the way, guys, did you notice that this week my virology course was featured on iTunes University.
Is that right?
Yeah.
I noticed it because you tweeted it.
That's right. That's right. It's a little self-promoting, but I do want people to learn virology.
That is my understanding of the purpose of Twitter.
That's right. So, yeah, it was cool. Apple featured my virology course on iTunes U. They had a big graphic on the front page for a couple of days. It might still be there. I did put up a photo at virustalk.posterous.com or Posterous. Anyway, I digress. You can also have an app to stream the episodes to your iPhone or Android device. You can find that over at microbeworld.org. And even though we are way behind on our email, we still love to get your questions and comments. Please send them to twiv@twiv.tv. Alan Dove is at alandove.com and on Twitter as Alan Dove. Thank you so much for joining
Always
us
a pleasure
today.
Yep.
And Rich Condit is at the University of Florida, Gainesville. Thank you, Rich.
Quite welcome. I love these email shows. They're some of my favorites. I think they're great.
Yeah, it's really good. I guess we should do them once a month. That would help.
Terrific listeners.
Yeah.
Terrific listeners.
Yeah, we have great listeners, and it gives us the opportunity to talk about so many topics. It's great.
Yep.
I think we have the best feedback of any podcast I have ever listened to. And if anyone disagrees, you'd send
then
me
they're
which
wrong.
one. Because I've listened to, yeah, right. I've listened to a lot, and I just don't hear the kind of feedback that we get. Really good questions.
Yep.
So thanks for that. I'm Vincent Racaniello. You can find me at virology.ws. And don't forget, everyone, if you want to read the tagline for TWiV, record it and send it in. Not too hard to do it. It's Matt Freeman did that last week. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.